Thank you. Good afternoon. My name is Atlang Gild Mpolokang from Botswana International University of Science and Technology. I'm here to give you a presentation titled Analysis of Exhaled Human Breath for Diagnosis of Tuberculosis Disease. So basically, we aim to develop a breathalyzer-like device for detecting of TB from exhaled human breath. And we focus on TB because, as you can see from this map here, it shows that TB has mostly affected the southern part of Africa, which include Botswana, where this research is based. And the TB diagnostic techniques, they include the Mantoux test and the sputum test. The challenge with this test is that they involve invasive procedures and they take time before patients can get their results. And as the patients are waiting for their results, the disease will be spreading. So because of these challenges, there is therefore a need for a device that is non-invasive, rapid, and can be used at a point of care. So what we know now from literature is that human breath, it contains volatile organic compounds. That is information about the metabolic process taking place in the body. Therefore, there are VOCs that can be produced because of the presence of the disease. So, and we also know that there are chemical-resistive gas sensors that can be used to detect these VOCs at low concentrations of parts per million. And the chemical-resistive gas sensors, they give they show a change in resistance. They show a change in resistance when they're exposed to these gases, as can be seen in this graph here. And these chemical-resistive gas sensors, they are mostly made from metal oxide semiconductors, because metal oxide semiconductors, they have high sensitivity, but they suffer selectivity. They have long response and recovery times. So studies have shown that when you encapsulate this metal oxide into carbon nanotubes, as seen in this research here, it shows that when you encapsulate the metal oxide into carbon nanotubes, you can enhance, you can tune the selectivity, you can shorten the response and the recovery time. So this is the study that was done by my supervisor in Botswana. So from this knowledge, we now can develop an electronic device that can sense the VOCs that are associated with the disease. And what we have, in order to do that device, we first need to identify and verify the biomarkers of disease. So for that, we are going to use a gas chromatography mass spectrometry, which is a GCMS. It works by a gas mixture is separated. It separates a gas mixture into individual compounds. So you're going to get a GC spectra with a peak. So each peak it represents an individual compound. And then as these compounds, they enter the mass spectrometry, where they are ionized and are analyzed using the mass analyzer. From there, which means each compound would have the mass spectra. So after identifying and verifying the biomarkers, we now synthesize the gas sensing material using various techniques like chemical vapor deposition, that's the CVD, and other techniques. From there, we build a sensor array and perform gas sensing measurements to obtain the response signals. Then we develop the machine learning models, which will be trained to understand the signals of TB and the signals of non-TB. From there, we can perform the trials where we are testing new breath to see if is it for non-TB or for TB. And what we have done so far, we've collected breath samples from TB patients and multi-drugs TB patients and controls. So the controls, these are individuals who did not show any symptoms of TB. And we collected the breath samples using a Tetla bag. So we collected the samples early in the morning before the patients could eat anything or take their drugs. And the samples were taken for 3 consecutive days. So from each patient, we were taken for 3 consecutive days. So we used a Tetla bag to collect that. And then from there, after collecting, we use a solid phase solid phase microextraction fiber, that is SPME, to pre-concentrate and collect the VOC from the Tetla bag and then characterize using the GCMS, which I've shown how it works. Then from there, we used an OpenChrom software to analyze the GCMS data and use the machine learning classifiers to classify it into TB and non-TB. And so moving forward to our results, so this is the GC spectra that we get. So we have the peaks here representing the compounds. And so this is for TB and the control sample, to give you an example. So from this, we can see that there's a difference in terms of the concentration and peak positions, which shows that between TB samples and the control samples. So this shows that there's a difference in the breath profiles of TB patients and the controls. So now, in order to identify these compounds, a peak is selected and then its mass, to Z spectrum, it is compared with the one in the NIST level database. So like for in this case, we had a peak from the breath sample, which matches tridecane in the NIST level database it matches by 92%. So this is an excellent match. But to further verify this peak, we ran a tridecane standard. And at the same retention time as the peak, the peaks were obtained at the same retention time. So also, its mass to Z spectra is 95%. I think match factor is 95%. So which means this is an excellent match. So this shows that our analytical method, or the method that we use to analyze this, is an accurate method. So yes, so this is how we identified those VOCs. So now after identifying the VOCs, we compared between the patients and the controls. And what we observed is that there are 8 volatile organic compounds which can be associated with TB. And among these 8, 6, they've already been identified, and they've already been, which are the unhighlighted ones. They have already been reported in literature before, so which means our study was able to verify the VOCs that have been previously reported. And for the first time, we report ocyanamide as a VOC that is associated with TB. And note that this ocyanamide was, there's a M-cyanamide, which has a similar structure as ocyanamide, was reported by Michael Phillips in a study from the Mycobacterium culture. So note that the Mycobacterium is a bacteria that causes TB. So for us, we found it in TB patients. So which means that the ocyanamide is due to the presence of Mycobacterium in the body. Also, we report benzene 1-methyl-4,1-propanol as a biomarker for multi-drug resistant TB. So note that this VOC we only found in multi-drug resistant TB and not in TB patients or in the control. So this shows that this VOC could be a signifier for that a person is now developing resistance to TB drugs. And from there, we also know that 4-methyl-octane, which is one of the VOCs that we also reported. So 4-methyl-octane has been reported in lung cancer, asthma, COPD patients, and children with TB infections. And because one of my colleagues is working on lung cancer patients, so we compared the concentration of this VOC in lung cancer patients and in TB patients. And you observe that on average, lung cancer patients has higher concentration of this VOC as compared to in TB. Okay. As compared to in TB. Then, so which means that because we see that lung diseases, they share some similar VOCs. So we cannot now say we diagnose a disease based on one VOC. We need to have a number of VOCs to use to diagnose the disease. And so from those results, we published we had a publication on those results in Nature and scientific reports. And we also have a second paper on lung cancer, also under scientific reports. Yes. And we do not stop there. Now, to build our array, we fabricated a microchip in the US. That's at M-Nano fabrication facility. So this is a picture of myself in WNF. So we fabricated a chip where we are going to deposit our sensing materials to sense the VOCs. So the non-conducting part of this chip is silicon oxide, which was deposited using plasma-enhanced chemical vapor deposition, and the gold was deposited using the sputtering technique. So because of time, move on. And to get the chip like this, we used photolithography technique was used. And so now that we have the microchip for making our array, so we are going to deposit our sensing materials on our array. Then we can sense the breast samples where we would obtain the signals for TB and the signals for the control. Then after getting these signals, we train our model to understand the signals of TB and understand the signals for the control. From there, now we can do the trials where we test new breath to see if it can tell if this is for a TB or this is for non-TB. Okay, a minute. So far we already designed a sensor for detecting triticane. So this is one of this paper also have been published. So for sensing triticane is one of the biomarkers that we have identified. We are now working on finding other sensors for other VOCs that are associated with TB. So this is what I'm currently working on at the moment. And that's it. Thank you. And these are my sponsors.